I have once forgotten to do that, and I haven't forgiven myself for it. So we are now recording. So just to remind everyone that we're recording Casey's lecture, and we'll be recording the questions afterwards. So as per usual, as we've been doing all year. So it's my great pleasure to introduce to you today Dr. Casey Fowler, one of our young guns in the Department of Biological Sciences. I'll give you a little bit of Casey's academic pedigree. I've just found out, I knew he was from Southern Ontario. I've just found out that he actually grew up in Kingston, and he did his first degree at Queen's, where he did an honors degree in biochemistry. And after that, Casey went to McMaster to do his PhD, where I think you sort of now you have sort of two things in your background. One's synthetic biology and the other one's microbiology.
That's exactly right. I had two supervisors as a PhD student.
Right. And that was as a PhD student, where those two things, you were working at the nexus of those two things, and they've sort of, they're important for your development as an academic. So after a PhD in McMaster, Casey spent a few years at Yale as a postdoc. That place called Yale. It's the University of Alberta of the south of the border. And what year was it? It was 2019 when we finally recruited you back to Canada. So Casey has had the sort of traditional Canadian academic career, where you start off with your undergraduate and graduate degrees in Eastern Canada, move outside of Canada to become a postdoc and really cut your teeth on some deep stuff where the funding for postdocs frankly is better. And like many of us, returns to Canada and moves west, which is another pattern that I'm actually familiar with myself. I'm also a Queen's grad, and I also ended up at the University of Alberta. So welcome, Casey. And with that, I'm going to turn the floor over to you to tell us a little bit about what you do and what motivates you and what kind of things we can look forward to coming from your lab. Casey, take it away.
All right. So thank you so much, Dave, and thank you everyone for coming today. So yeah, this talk was actually originally scheduled for April 2020. And then of course, COVID hit, and here we are. I'm finally giving the talk a full year later. So, yeah, so if the talk falls flat today, I guess I can't use the old, I didn't have enough time to prepare excuse, right? So, but yeah, so here I am finally giving the talk. I've had a chance to listen to a number of the other chairs lectures, and I've really been enjoying them. And one of the things that I've really enjoyed about the chairs lectures is that the talks are all very different, right? They're very different in part because the disciplines are very different, and also sort of the different approaches people have taken. And the approach that I'm going to take today is sort of a big picture, sort of concept-driven talk. So sort of in contrast to what I would sort of call a normal talk that I would give, maybe a little less data-driven and a little sort of more big picture-driven. There'll be a little bit of data sprinkled in certainly throughout the way, but the idea here is to sort of get across what I do. And I think that maybe makes sense for a few reasons, one of which being that it is a very diverse department. It's one of the great things about our department, and so I think the big picture sort of works better for that. But also, I don't know a lot of you. There's a lot of people out there I haven't even really had a chance to meet. COVID hasn't helped that. And even those that I do know maybe don't know a lot about my research program. So the goal today is to sort of communicate with you guys a little bit about what it is, some of the things that we're interested in, and some of the things that we do. So as you can probably tell from the title, my lab is interested in Salmonella. And one of the nice things about studying Salmonella is that most people, just your average person sort of off the street will have heard of Salmonella. So I often, ask the question, what comes to mind, when you hear Salmonella? And I actually still remember the first time I heard Salmonella, or at least the first time I processed it. And I remember asking my older brother what it meant. We were watching The Simpsons together. And it was the episode where Homer eats like the tainted hot dog from the convenience store. And anyway, so I remember like hearing that word and asking my brother like, oh, they use the word salmonella. I asked him what salmonella is. So maybe that's what comes to mind for you. You're thinking of The Simpsons, or maybe you more think of, danger, what not to do. Why this is a spectacularly bad idea. So this is French soccer player Patrice Evra, who I think several years ago now posted this video on Instagram or Twitter, I'm not sure, of him like sucking on and licking a raw chicken. I never figured out why he did this. And I think sometimes when you kind of encounter something like this, it's better not to look too deep. It's sort of like a better story in my mind, just sort of seeing him do this without a real purpose. I can't fathom what the purpose would have been. But anyway, you know, when you think this, you think, oh, that's a terrible idea. And if you ask somebody why, maybe one common answer they would give you would be, you know, salmonella, right? Maybe that's not what you think of. Maybe you think of your younger days when you used to, you know, go to your favorite pub on Wednesdays for 10-cent wings and not maybe question, you know, why the wings were only 10 cents and you remember maybe paying the price the next day. Or maybe you remember back in 2017, right here in Edmonton, when you went to the old folk fest, the Edmonton Folk Fest, and you visited the wrong food booth. But, you know, regardless of, you know, where on that spectrum you lie, odds are the same thing probably comes to your mind, and that's food poisoning or gastroenteritis, which is for good reason. So salmonella is one of the sort of leading causes of food poisoning. So salmonella gastroenteritis is sort of the food poisoning you get is sort of what you think of, and it's variable in terms of how intense it is, and the symptomology can vary a little bit, and I'll talk a little bit about that later. But generally speaking, it's sort of the food poisoning you think of, right? Sort of a self-limiting diarrheal disease where you, if you're a healthy adult, will last a couple of very unpleasant days, and then you'll clear the salmonella. If you're a vulnerable population, such as a young child, an older adult, or immunocompromised in some way, you can have some more sort of severe side effects that can come even from your run-of-the-mill salmonella food poisoning. It's very common, so it's difficult to estimate the numbers actually, because most people don't go to the hospital, but it's estimated that the CDC in the United States estimates somewhere in the vicinity of about 1.4 million cases per year. In Canada, you know, on a per capita basis, those numbers are probably pretty similar, and salmonella food poisoning is a pretty major issue sort of around the world. It isn't a North America problem or even a regional problem. It's a big problem everywhere. And it's, of course, got a massive, global health impact and also a massive economic impact, right? So depending on how you estimate that impact, it's obviously clearly in the billions per year. It's got a huge impact on the agriculture industry as a foodborne pathogen, and so it's a major problem. When we say salmonella, we're talking about the bacterial species, Salmonella enterica. So it's a bacterium. There are viruses that can cause food poisoning as well. Salmonella is a bacterial cause of food poisoning. For the microbiologists listening, it's a gram-negative proteobacterium, and really they make their way in the intestinal tracts of animals, right? That's the sort of key environmental reservoir. They can survive sort of out in the open world, out in the environment for extended periods of time, but where they really sort of thrive is in the intestinal tracts of animals. And sort of globally, sort of as Salmonella enterica as an overall species are able to infect, or in some cases probably colonizes is a better word. It may be behaving more as a commensal in certain instances, which I'll actually touch on later as well. A very wide range of animals. So warm-blooded animals, cold-blooded animals. And so it's very sort of evolutionarily a very successful bacterial pathogen, and it's also a very diverse bacterial pathogen. So there's different ways you can look at diversity, and bacterial defining species and subspecies and this and that can be sort of tricky and complicated, and Salmonella is no exception to that. In Salmonella, we often talk about serovars, or serological variants, essentially identifying the Salmonella on the basis of their sort of some molecules on the surface of the cell that we can detect. So that's sort of the typical way that they get described, and we know of more than 2,500 different serovars out there, and in many cases those serovars can be very different from one another. And so that's actually a big part of the reason that my lab is so interested in Salmonella, is this tremendous diversity in this bacterial pathogen. So why do we study Salmonella? Two sort of like big picture reasons. One is it's really useful as a model system to sort of understand biology, right? So we're really interested in host pathogen interactions, and Salmonella is a great model system for that. There's a number of reasons for that. One reason is that we know quite a bit about Salmonella. Salmonella are easy to grow. Salmonella are, relatively speaking, easy to manipulate genetically, and we have some great infection systems, so infection model systems, so cell culture systems or animal model systems to study Salmonella. So it's a great model system for host pathogen interactions. And it's also a great model system, I would argue, for understanding sort of diversity and evolution in bacterial pathogens, which is sort of the major theme of my lab, because of all of this diversity and the fact that it's so useful as a model system for studying host pathogen interactions. Then you have the sort of more practical reason to study Salmonella, which is to learn about Salmonella because it's a really significant global health problem, right? So, you know, because it's such a significant global health problem, there's really a need out there for different, for sort of new and improved ways to either diagnose Salmonella infections, detect Salmonella, or to prevent or to treat Salmonella infections. That's also something that my lab is interested in. All right, so I'm talking about this Salmonella diversity, and it's the naming and the lineages and stuff can be very complicated, but I want to just sort of highlight a few things here. So I mentioned at the outset that we all know Salmonella for causing food poisoning, and not all of those, you know, not all of these 2,500 serovars, first of all, can even cause an infection in humans, right? So many of these don't infect humans at all. Of those that do, there's significant differences in terms of their ecology, so the different types of hosts, right? So these things often infect a range of different animals. Many of them are broad host range, so they'll infect a very broad range of, in some instances, both warm and cold-blooded hosts. You can have very narrow host ranges or even host-adapted lineages that'll only infect like a single host. There's human-adapted Salmonella out there that I'll talk about in just a moment, for example. So you've got a lot of diversity in terms of the hosts, how broad the host range is, what that host range is. Also in terms of the virulence, right? If we're thinking about human infections, so how likely their propensity to cause disease in humans, but also the nature of that disease, the symptomology, the types of disease that they cause. There's a lot of variation there. And one thing that I'll highlight here, which is sort of interesting and doesn't really get thought about a lot, is that most of the Salmonella that we're interested in are in this one. So we have Salmonella enterica as the species. What we're mostly interested in. There's another Salmonella species, same genus, different species, called Bongeri. And then within the enterica species, there are 6 different subspecies. And most of what we focus on is over here, this enterica subspecies. And there's a lot of diversity and a lot of interesting serovars in here. And this is what we mostly focus on. But 5 of the 6 different subspecies, as well as the other sort of major Salmonella species, are all more adapted seemingly to cold-blooded animals. We pretty much only isolate these guys from cold-blooded animals like reptiles and amphibians. And in fact, reptiles and amphibians have really high carriage rates for Salmonella. So if you just pull a reptile or an amphibian out of the wild or from a zoo or a pet store or for somebody's house, and you sort of, there's lots of studies out there that sort of ask what percentage of reptiles are infected with or carrying Salmonella, maybe is a better way of putting it. You get really high incidence rates. You can find multiple different serovars being isolated from the same animal, and so it seems, and often in the overwhelming majority of these cases, the reptile, for example, is showing no signs of illness, and so it seems like, in contrast to sort of the way that we interact with Salmonella, in many cases in reptiles, for example, they might be more of like a commensal that's lingering around in their intestinal tracts. And that this evolution of these, you know, mammalian adapted or warm-blooded animal adapted lineage, they may have taken sort of on more of a virulence pathway. But it's interesting that some of these cold-blooded guys can actually cause disease in humans too. Usually the disease is limited to sort of, again, those vulnerable populations that I spoke about earlier. So young children, for example. There we see a lot of differences in these species, right? So we can find that certain of these have a hard time infecting sort of a non-vulnerable person, but when they infect a vulnerable person, some will cause a really mild disease and others will be sort of a much more dangerous infection. So this high carriage rate in reptiles actually is the driving force behind a lot of the laws that are in place that dictate, you know, your ability to buy a turtle, for example. So in the 1970s, the US government put, for example, there's other laws, other places, put rules in effect about being able to, the types of turtles and who can sell what types of turtles, essentially, because turtles were causing huge numbers of Salmonella outbreaks. Actually, if you go to the CDC website, they have this like adorable video. I'm just showing you called The Trouble with Turtles, where they have this like little kid voice narrating the dangers of turtles and what you should and shouldn't do with the turtle. Anyway, if you're interested, you could go check that out. I won't take the time to show that today. But this still causes significant outbreaks today. So this is an article from Global News. Stop kissing pet turtles, the CDC warns. Salmonella outbreak hits 13 states. And it's actually estimated by the CDC that more than 5% of all salmonella cases come from interacting with reptiles. And so more than 70,000 cases per year from the CDC. You get different estimates on this, but it's clearly still a significant way that people can become infected with salmonella. And again, this is a much more severe issue with the vulnerable populations. So I know it really seems like it's probably a good idea to let your young children play with your 19-foot python, but make sure that when they're done, they do a good job washing their hands, because it turns out that they're a source of salmonella. All right, the last sort of diversity thing I'll talk about is maybe the thing I'm most interested in, which are these human-adapted lineages that I mentioned earlier. And so human-adapted salmonella cause a very different disease than the food poisoning that we discussed earlier, right? So Salmonella typhi is the key serovar here. There are some other serovars like Paratyphiae that are sort of similar but a little less sort of common. And these cause very different disease. These cause, instead of a lot of intestinal inflammation, these keep a lower profile in the intestines. They spread to systemic sites and cause these prolonged systemic infections that, if untreated, are life-threatening, particularly in children. So children in the developing world, this is a massive problem. And the disease here is called typhoid fever. So this is a fundamentally different disease than the gastroenteritis that we spoke about earlier. And so the virulence properties and the virulence traits of these typhoidal serovars are very different from those of the non-typhoidal serovars. And this despite the fact that genetically speaking, they're quite similar. And that's something that we and many others are very interested in. And so because these are human adapted serovars, they can't spread to us from animals, right? They spread from person to person, typically via a contaminated drinking water intermediate. And so the reason that we all thought of food poisoning when I said salmonella, and none of us probably, or very few of us at least, had typhoid fever even pop into our minds, is that typhoid fever essentially doesn't exist anymore. It's at least not endemic in any countries in the world that have in the developed countries that have ready access to clean drinking water. And that's because the Salmonella typhi spread from person to person via the fecal oral route. And that's typically involves fecal matter, either sort of directly or indirectly ending up in the drinking water supply. So ending up in well water or something like that. And that being a really important way that the bacteria spread from person to person. And if you have good sanitation and ready access to clean drinking water, you can actually eradicate the disease. But unfortunately, in many places of the world, this isn't the case. And typhoid fever is a massive problem. So estimates are in the range of 20 million cases per year and 200,000 deaths per year. Another really interesting feature of these human-adapted serovars is that about 2% of the people who become infected with Salmonella typhi become persistently infected. So they'll carry the bacteria usually asymptomatically, so they're not showing any signs of infection, but they'll become what we refer to as carriers. And they can be carriers for weeks or months or years or even for life. And so there's some well-known sort of historical figures who are characters. You may have heard of Typhoid Mary, for example, who are known to have spread typhoid fever to huge numbers of people because they are carrying the bacteria, and they'll sort of shed these bacteria periodically in very large numbers, and that can initiate outbreaks. And so these carriers are thought to be, sort of evolutionarily speaking, a really important environmental reservoir for these human adapted serovars. Okay, so in addition to all this diversity, another reason these Salmonella is so great for studying right now is that there's this incredible wealth of genomic sequencing data. So, I think this is true for everyone, basically in biology, where at one point, genome sequencing didn't exist, and then it became a little easier to come by, and that continues to improve. But I would argue that for Salmonella, there's just an embarrassment of riches now. And that's been driven largely by the fact that the cost of whole genome sequencing has gone down so much that a lot of public health agencies are actually using that as a way to, when they get an isolate, as a way to sort of figure out what that isolate is, rather than just doing serotyping. They'll actually sequence the whole genome and deposit this genomic sequencing data. And so this enormous amount of sequencing data allows us to make some really interesting comparisons, right? If we're really interested, so my lab's really interested in virulence factors and genetic factors and sort of the molecular level differences that cause different sort of phenotypes and disease traits. And we can make these comparisons either at the species level, the subspecies level, serovar, even individual strain level. What's going on genetically with these species that could be causing these differences? Or if we get really interested in a set of virulence factors, where are these virulence factors? Where aren't these virulence factors? There's a lot of tools at our disposal that we try to make use of. So the Fowler lab, maybe in a nutshell, exploring the diversity and evolution of Salmonella virulence. That's a massive, sphere of things you could study. And of course, we've had to narrow it down a little bit more than that. And I would say that there are basically two major lines of research going on right now in the lab as we're getting up and running. The first line of research, or one of the lines of research, relates to understanding how horizontally acquired genes get plugged into regulatory networks in Salmonella. So, point mutations and accumulations of mutations and things like that have certainly contributed to this diversity in Salmonella. But the real driving factor seems to be horizontally acquiring genes from your environment, or degradation of genes, like sort of gaining and losing genes. And when you gain a new gene that might give you a new interesting virulence phenotype, we're really interested in understanding how that gets plugged into the biological program. So specifically about understanding how that gene gets regulated in the way that it needs to be regulated, because it's really important for new genes, and especially virulence genes, to be turned on and off to the right levels at the right time. And so we're really interested in understanding sort of the evolutionary aspects of that and how that process works. And I'm not going to talk about that today. If I'm lucky enough to be invited back to give another chair's lecture down the line, maybe I'll talk about this next time. Today I'm going to take the sort of second half of the talk here and tell you a story about the evolution and diversity of AB5 type toxins in Salmonella. All right, so AB5 type bacterial toxins are a really important class of bacterial virulence factors. And so what they are is they're bacterial proteins that the bacterium makes and produces, and they form these complexes. So I've sort of shown that schematically here. And after they produce these complexes, they secrete them. So they secrete these toxins out in the context of an infection. And when these, so when they're engaged with a host, and when these toxins are secreted, they're able to actually bind and in many cases target specific types of host cells. So if we're talking about a human infection targeting human cells, they can bind specific types of cells and gain access to those cells, and then they can go in and they can manipulate the biology of those cells in some way that ends up being beneficial to the bacterium, and oftentimes to the detriment of us or of the host. And so they're called AB5 bacterial toxins because of their architecture. They're made up of two different types of subunits, the A subunits and the B subunits. And even though there's quite a bit of diversity in these toxins, which I'll show you in just, I think, the next slide, they're all sort of put together the same way. We all have this A subunit that's sort of inserted in the middle of this sort of donut-shaped pore of B subunits. This is the AB5 architecture. Again, even with toxins that have little or no sequence homology, you'll still see the same basic architecture. So the two different types of subunits, the A subunits are what are known as the active subunits. This is where all the glory is. These are the subunits that actually exert the biological effects of the toxin. So typically here, this is an enzyme. And so this would be, for example, an enzyme that will go into the host cell and target some very specific aspect of the cell. So it'll target, for example, a very specific protein in the host cell, and it will maybe modify that, it'll covalently modify that protein in such a way that it affects the way that protein works. So it'll degrade the protein, it'll chop it up, or it doesn't have to be a protein either, as you'll see later in the talk. So it's exerting some very specific biological effect in the host cell. The B subunit, also very interesting, also essential for the toxin, but maybe not quite as much glory, right? This is the unsung hero that actually gets the A subunit where it needs to get to. So this mediates the toxin trafficking and delivery and specifying the types of host cells that it's going to enter. And so these B subunits bind, for AB5 toxins, bind glycans on sugars essentially that decorate the surface of host cells. So there are a lot of prominent AB5 toxins out there. This isn't all of the AB5 toxins, but these are probably the three best known. So we've got cholera toxin, Shiga toxin, and pertussis toxin. A few things I want to highlight about AB5 toxins. One is the sort of diversity that I mentioned earlier. So for example, if we compare cholera toxin and Shiga toxin, their B subunits have some similarities in their folds, but they're not related in any way, detectable way at the sequence level. And their A subunits are just totally different, right? They're totally different enzymes, different folds, different sequences, and yet they adopt this similar structure. And that's sort of interesting, and I'll come back to this later in the talk. The other sort of thing I want to get across here is that these are really important virulence factors. So the three that I have on the screen right now are crucial virulence factors for really important human diseases. So cholera toxin, for example, is thought to be directly responsible for the sort of key symptoms that we associate with the disease cholera. And if you don't have this toxin, it's thought that Vibrio cholera, the pathogen that causes cholera, can't cause the disease. Similarly, Shiga toxin is found in a few different places, most notably probably the STEC, so the most dangerous E. coli, the E. coli we often hear about in the news, the O157H7. So Shiga toxin is their most sort of important virulence factor. They have others, but it's thought to be directly responsible for the most severe types of disease that are commonly associated with E. coli O157H7 infections. And then pertussis toxin, again, is a central virulence factor for pertussin, and this is perhaps demonstrated well by the fact that it's actually, we don't hear much about pertussis because we have a good vaccine against it. We all get the Tdap or DTaP vaccine, and the P in there is for pertussis, and actually an inactivated version of pertussis toxin is actually a really key component of this vaccine. And in fact, they aren't AB5 toxins, they're AB toxins that don't have that same conformation, but the other two sort of main components of the Tdap vaccine that protect us against tetanus and diphtheria are also inactivated versions of toxins. So sort of highlighting the important role that bacterial toxins play in disease. Okay, so I mentioned that Salmonella have some of these toxins. This was actually went on unidentified for a very long time. So for the first, well, some toxins, were identified even if we didn't know everything about them a very, very, very long time ago. There was a solid, 100 years of Salmonella research before the fact that they encode AB5 toxins was recognized. And so what really caught the eye right away about this was that this putative AB5 toxin that's now a bona fide known AB5 toxin or related to an AB5 toxin, I'll get to that in a second, is encoded by these human adapted Salmonella, but generally speaking, not encoded by the broad host range serovars, the food poisoning serovars that we're most familiar with. And at the time, actually, that this toxin was discovered, it was thought that they were only encoded by the human adapted serovars. So it was thought, and this was really exciting, that this mysterious toxin may play some really important role in sort of the unique virulence features of the human adapted pathogens, of the human adapted serovars. And so we've got this locus that's present in the Salmonella typhi genome, and this is the same locus in the Salmonella typhimurium. This is an example of a food poisoning Salmonella. And so as you can see, it's got these unique genes here, these PLTA and PLTB. PLT stands for pertussis-like toxin. It's sort of a distant evolutionary cousin from pertussis toxin. It's quite a different toxin, but it's evolutionarily, it's related. And so we've got this AB5 toxin here was sort of what was originally thought, but it was intriguingly eventually discovered that this in fact isn't an AB5 toxin, it's actually an A2B5 toxin. That this locus also encodes another actually known toxin subunit. It's an active subunit of a different toxin called cytolethal distending toxin. And this active subunit essentially hitches a ride with the AB5 toxin. There's this unique cysteine residues in both the PLTA and the CDTB relative to sort of their other evolutionary cousins that allow them to form this disulfide bond that allows CDTB to become covalently tethered to PLTA and sort of hitch a ride along with PLTA to form this sort of double active subunit toxin that we call typhoid toxin. And so the structure of typhoid toxin was solved, I guess about 8 years ago now, by a really talented structural biologist named Xiang Gao, who at the time was a postdoc, but now has, for several years now, actually has had his own lab in China. I overlapped with him as a postdoc, and he's actually a collaborator and a friend of mine. And so I'm hoping to exploit his tremendous structural biology skills to help us out with some toxins that we're interested in down the line. But regardless, he solved the structure of typhoid toxin. So this is what it looks like. And so these are the color coded here are the 3 different types of subunits, right? So we've got the I had this schematically on the last slide. We've got the CDTB. This is one of the active subunits. It's a DNase. It gets into the nucleus of the host cell and introduces double-stranded breaks in the genomic DNA. This DNA damage, depending a little bit on the extent of the damage and the cell type, elicits either G2M cell cycle arrest, essentially inactivating the cell, or in some cases, cell death. The other active subunit, PLTA, is an ADP ribosal transferase, much like pertussis toxin, but the actual sort of physiologically relevant host cell target is unknown. We know it's an active enzyme. We know it can do this. We think it's probably got a really important biological role, but it's not known what the target of the ADP ribosal transferase is yet. So all of the phenotypes we can associate with typhoid toxins seem to map back to this DNase, which seems to sort of be the dominant phenotype, at least, that we can detect. And then the delivery subunit, the B5 part of the toxin, is a subunit called PLTB. We've already talked about that. And this is where, of course, the glycan binding comes in, right? The toxin delivery. Binding those host cell receptors and triggering the uptake of typhoid toxin into target host cells and getting those A subunits where they need to get to. So in the decade or so that we've known about typhoid toxin, there's been a number of different studies trying to get at its role in typhoid fever and Salmonella typhi pathogenesis. And this has been done a number of different ways. So this is sort of a big picture overview of some of the ways that people have tried to get at this. Either looking at purified toxins or infection models where maybe you've made mutations in certain toxin genes, or either deletions or point mutations or different things. And then we look at how these things will interact or how these things will work either in a mammalian cell culture model or animal models. And sort of the bottom line of these things is that there's really some pretty strong evidence at this point that this is a really central virulence factor for Salmonella typhi. This toxin actually isn't produced if you just grow Salmonella typhi in normal laboratory media at all, which is a big part of the reason this toxin went undetected for as long as it did. But when Salmonella typhi engage human cells, this thing gets produced at very high levels. Okay, so this is clearly associated with human infection. It's very potent, so even at relatively low sort of moieties of infection, low in doses of infection, you can inactivate virtually every cell in the cell culture dish. So this is a very potent toxin. And if we put this toxin into laboratory animals, interestingly, you can actually recapitulate, you can actually elicit many of the symptoms that are actually associated with severe typhoid fever, suggesting that the toxin may actually be playing some direct role in eliciting some of the symptoms of the disease. And so the full role of the toxin in Salmonella typhi pathogenesis isn't completely worked out yet, but the prevailing model that I think anyone I know who's working on typhoid toxin sort of thinks is what's going on is that this is probably important for Salmonella to either get to the site that it occupies during persistent infections, which is typically the gallbladder, or to remain there. So essentially for establishing that carrier state, for establishing these sort of long-term persistent infections. That's what we think the toxin's probably doing. It's probably the evolutionary driving force for having this toxin. Okay, so as a postdoc, I was actually working on some other aspect of the biology of this toxin, and it was becoming apparent as more and more genomes were being published that this toxin wasn't just limited to the human adapted serovirus, right? That as more and more Salmonella genomes were popping up, it was becoming apparent that this toxin was present elsewhere as well. And so I wanted to, as sort of a side gig, I was looking at where this toxin is and isn't and trying to get a handle on the sort of distribution of the toxin. And when I was doing that, I stumbled across the fact that in the Salmonella typhi genome, so elsewhere in the genome, not at the same locus as the rest of the toxin genes, but elsewhere at a distant location in the genome, there's actually a PLTB homolog, a pretty distant homolog. It's only 23% identical, but statistically speaking, a very significant homolog of PLTB. So another toxin B subunit was present elsewhere in the typhi genome. And this locus had actually been noticed before, but it had essentially been disregarded because it's essentially a minefield. It's littered with what are known as cytogenes, which are sort of DNA sequences that used to encode a protein, right? We know that because they're homologous to proteins that we know about, but they've been inactivated, they're degraded. So for example, the start codon will be missing, or there'll be a stop codon a few codons in, or something like that. And so we have lots of cytogenes, including an A subunit immediately upstream of the B subunit. And so on its own, you wouldn't necessarily think too much of it, right? The A subunit's inactivated. This may not be expressed at all. There's no reason, it may just be intact because it just hasn't happened to accumulate high. And so this is, I won't go into the details of this, but this is actually closely related to another type of toxin, another AB5 type toxin called ART-AB. So there's a degraded relative of another type of toxin, a related type of toxin elsewhere in the genome, where that B subunit is intact. And so when I was looking around at all this sort of typhoid toxin distribution stuff, I discovered that typhoid toxins sporadically distributed in different lineages, different subspecies, and sometimes even different on the strain level. So I was interested, okay, well, if we've got this B subunit, is this just a fluke of Salmonella typhi, or do we find a second B subunit elsewhere sometimes as well? And so this is, the answer to that is yes, that essentially where there's a typhoid toxin, there's a second B subunit. So this is the typhi scenario that I just had on the last slide. I'll just show you, there's many, many examples of this, but I'll just show you a few. This is the other species. So in the Salmonella genus, Salmonella bongeri, they actually have typhoid toxin in roughly the same place that Salmonella typhi does. The sequence is quite different, but it's still there. And elsewhere, totally different spot in their genome. And actually it doesn't even look that much like this one, but there's also a second, what we'll call orphan B subunit, a B subunit without an A subunit, basically. In the Di-Arizona subspecies, the typhoid toxin islet actually has two B subunits back to back. So we have this time the second B subunit's right at that same locus. And maybe my favorite example is you can actually find Salmonella typhi, or typhoid toxin, excuse me, on prophage sometimes. Essentially viruses that have invaded bacteria and their viral genome is now incorporated into the bacterial genome. And this weltivrate and cerevar, generally speaking, doesn't have the toxin, but it's got this prophage, and so this specific strain has the toxin. And elsewhere, it's got another prophage that just has the B subunit, which I think is pretty cool. And so you never find the orphan B subunit without a typhoid toxin locus, and you virtually never find a typhoid toxin locus without a second B subunit. So to me, something had to be going on here. And our hypothesis was this, that this was the toxin that we knew about. This was the toxin I've shown you the structure of. We thought that maybe this was going on, right? That this second B subunit could actually form an alternate toxin, or perhaps that it could interact with that existing B subunit to form these sort of heteromeric delivery platforms. And so we went about testing this. We first checked to look at the expression, and we saw that this gene here, like these other genes, was specifically expressed when Salmonella typhi engages with human cells, invades human cells. So we knew that it was co-regulated, and so we decided to check it. That was sort of a really positive sign. We thought, okay, this gene's actually being expressed, and it's being expressed at the same time as the rest of the toxin genes. So does it actually form a complex was sort of the big key question, and does it form a complex during infection? And the answer to that, to cut to the chase here, is yes. So we looked at this a number of different ways here, and I'll just show you one panel without the controls of data, showing that if you just pull down, if you grow Salmonella typhi under conditions where you produce lots of this toxin, including infection conditions, this is actually in vitro, but, and you pull down with an antibody that's directed against PLTC, an epitope on PLTC. So this additional toxin gene, the second B subunit, which we're calling PLTC. If you pull down on that and you ask the unbiased question, what comes with it? So you look at it by mass spec and just say, does anything else sort of significantly come down with this? You see that CDTB and PLTA come down sort of consistent with this complex formation here, and that we don't pull down PLTB, that other B subunit, at all. So we don't see any evidence for this. So again, we did numerous other controls and actually looked at this in other ways beside this mass spec way. But the sort of bottom line answer to this question is that this heteromeric toxin, we see absolutely no evidence for this forming, but we can see that both of these toxins are being produced by Salmonella typhi upon infection of human cells. It's producing both this original toxin that was identified and this other toxin that's got the same A subunits, the same two A subunits, but it's got this new B subunit, this different B subunit. And so we wanted to understand sort of some of the properties of this new second toxin. We knew quite a bit about the first toxin. We tested that in a number of different ways. I'll just show you a couple highlights here. So one thing that we did is we purified this second version of the toxin, and we applied it to cultured human cells. And then we looked for cell cycle. We did a cell cycle analysis to look for that G2M cell cycle arrest that I mentioned earlier, that's produced by that CDTB's enzymatic activity, the DNA damage that the toxin causes. And so we find that both toxins are quite good at initiating this G2M cell cycle arrest in these human epithelial cells, these cultured human epithelial cells, but that the originally identified version of the toxin, at least against this specific cell line, is more potent. We also looked at these two different toxins in animal models of infection. And again, this had been done with the original toxin, so we were interested in how the new toxin would behave. And so when you administer, what we're looking at here is 2 micrograms of purified toxin to lab mice, you actually get all the mice dying within actually exactly, in this experiment, 5 days post-toxin administration with the original toxin. And before they die, they start to show a range of symptoms, and what I'm showing you here is weight loss, so they start to lose weight, and then they die at 5 days post-toxin administration. The PLTC toxin, sort of like the results I just showed you, seems to be just a little less potent, right? You do get some death, but it's actually takes quite a while for it to happen. So it wasn't until about 10 days post-toxin administration that we start to see these animals die. Not all of them die, and those that don't die, they lose weight, but then they start to recover, essentially. So it just seems to be a little bit less potent. But interestingly, at these same doses, we see an increase in the ability of this toxin to deplete white blood cells, to essentially deplete the host circulating immune cells. And so that's one of the sort of symptoms of typhoid fever that is recapitulated by this toxin administration, is depleting these, the white blood cells, these immune cells. And the PLTC version of the toxin, even though it's not as potent, seems to be better at that. So we think that there are some really interesting differences in the way that these two different toxins behave, even though at this point we don't really fully understand them. And so I didn't show you most of these data, but we've looked at these two different toxins in a number of different ways, and there seem to be a lot of really significant differences between these toxins, right? In terms of the way that they're trafficked, in terms of the way that they behave in mice. They're both turned on in the same conditions, but they seem to be regulated, but they're definitely regulated by different regulatory networks and in response to different cues. And so we think Salmonella typhi is actually producing more or less of the different toxins depending on what cues it encounters. So we think that there's some really interesting biology here. But we're also really interested in the fact that these two very different B subunits, 23% identical, can both form a structure with that same A subunit, right? That's really interesting to us. And I think it speaks broadly to the fact that there's this really broad class of diverse toxins that have that same architecture. So we're calling this Lego-like assembly. And so if you think about Lego, you can get very different pieces, right? And those different pieces can still come together because just that little part that connects, that connection scaffold, is conserved in these different pieces. And we think that that's going on in these toxins as well, right? We've got these very diverse toxins. These toxins, if you actually look at the individual A subunits or B subunits, they're very different. But the sort of global way that they come together, this pore in the middle here, and this sort of peg that inserts in the middle, it's sort of an interaction of alpha helices from the B's and the A's, sort of guides this Lego-like assembly of these toxins. And that's why we're able to see these, very different toxins, both B subunits, both forming a toxin with those different A subunits. And we think that this may have fueled the sort of expansion of this AB5 class of toxins. And so in terms of some future directions here, as I kind of wind down, this is scratching the surface of interesting biology and interesting evolution of toxins in Salmonella. So if you dive deep on the bioinformatics of these toxins, you'll see that there's just tremendous, crazy levels of diversity. So this is showing you, I don't expect you, of course, to take all of this in, but if we just sort of focus on the last sort of column here, each line here is a different combination of toxins that can be produced by different lineages. So you just have an incredible diversity of different types of toxins that have these same, ADP ribosyltransferase transferase and DNA degrading activities that are being delivered in these different ways and in different combinations by these different lineages. And so we want to dig a little deeper in on this and understand this a little bit better. And we think that this relates to disease, right? So one question you might have is, if we've got these typhoid toxins and all these random, other lineages, do they all cause typhoid fever too? And the answer to that is that no, they don't. But we think that they probably cause more severe disease on average. And so this is some data that I did. I'll zoom through this because I'm getting late in the talk here, and there's a couple of things I quickly want to talk about. But I dove into some CDC data 30 or 40 years of data from the CDC, and I plotted essentially how often different serovars are isolated from systemic sites, which is a sign that these bacteria have spread beyond the GI tract and have caused a more severe infection. And I plotted that versus whether or not these serovars typically have a typhoid toxin locus or they don't. And what you can see is these black bars, these black dots, there's a very strong correlation between, a clear correlation here between the lineages that have the typhoid toxin and their propensity to form these systemic infections. And so, you know, this certainly isn't proof that this toxin's responsible for this, but this is, I think, really an intriguing thing, and it's sort of consistent with what we know about the biology of the toxin. So a little bit of data from my own lab, one slide here by a talented fourth-year student I had who's unfortunately leaving me pretty much now, Eric Schultz. A project that we're really excited about. Diving deep into the bioinformatics of these toxins, we think we've found a few different, what I call hybrid toxins, where you've got an A subunit from one class of toxins in a genome sitting right beside a B subunit of a totally different class of toxins. Really interesting, right? Sort of the missing link of how these toxins evolved, you know, maybe not directly, but some evidence of how that would work. So he's been working on something that I, you know, sort of found through these analyses, where we've got the A subunit, essentially, of Shiga toxin. It's not exactly the same as the Shiga toxin that you'd find in 0157H7. It's a relative of it, encoded immediately upstream of a B subunit that looks a lot like cholera toxin's B subunit. And so we think this is forming what I'm calling these hybrid toxins. And so Eric started working on that, and just the one piece of data I'll show you that suggests that this maybe is happening is we're moving this into a heterologous host, and we're overexpressing these two subunits back to back, and we're only tagging that B subunit, and then we're pulling down on that tag, so it's a his tag, and we're using a nickel column to purify out. And when we purify out the B subunit, the A subunit comes too, sort of a piece of very preliminary data suggesting that these are indeed forming a complex. So he's actually done some more control since then, and we think that this is real, and we're pretty excited about this. And I don't know that I really have time to go over this, so maybe I will just skim past this, unfortunately. Sorry, Luke. But we're not only interested in toxins, so we're also interested in other differences between different lineages and hunting through genomes. And so I had he's still in the lab. He's going to stay around in the lab as a graduate student, which we're really excited about. Luke, who's been a 398, a 499, and then a graduate student in the lab, did this project looking at an interesting genomic islet in Salmonella typhi that seems to be specifically for those typhoidal serovars. And we did some bioinformatics that suggests that this island is involved in sort of interbacterial competition. We think that this might be a bit of an immune system that Salmonella typhi has to protect it from perhaps the human microbiota. So we think that's neat, and we're going to do some experimental follow-up of this eventually as well. So this is our first paper out of the lab that came out within the last year. So that's we're excited about that, of course. And with that, I will end the talk, I think, basically on time here, and make some time for questions. I do want to acknowledge some people. So some of the data that I showed was from most of the data that I showed was from my postdoc lab. So I want to acknowledge some people there that helped me with that project. Jorge Glon is a phenomenal supervisor to have, really brilliant scientist, and I really enjoyed my time there. My new lab, so I showed very quickly some data from a couple of these students, and Ada and Adam, I'll try to show some data from them next time. They're graduate students who've started in the lab. And I also, of course, want to acknowledge some important funding sources as I get up and running here. So all right. Thank you for your attention.
Wow, thank you, Casey. I'm sure everybody else will join me in giving you a virtual hand of applause here. Fantastic talk. And let's throw the door open for questions. And a reminder, you can either speak up, or you could raise your blue hand or type something into the chat. There's a number of ways we can get our attention.
I'll ask a question, Casey.
Hi.
Great talk. That was really cool. So I used to work on AB toxins in grad school, but I haven't kept up with that field, and that was longer ago than I care to admit. So I didn't realize you could have that the binding subunit is really interesting, but you can also apparently have different A subunits attached together. So you have two different A subunits that are just connected by a disulfide bond.
Yeah.
Did I understand that right?
You heard that right. Super cool, right?
Yeah.
So
It
that's,
almost seems like the type 6 system, right? Where they kind of weaponize that injection device with different weapons. And so I was wondering, do you always see, you showed in your diagrams that you always have those two A subunits joined together. Do you ever see kind of two A's or you know, two of the red one or two of the blue one, or
yeah, they get combined in interesting ways. So I think that, so we don't see any examples where we have two of the, I think that the evolution that happened was that a unique cysteine popped up in one of these A subunits, and a unique cysteine popped up in the other A subunit, in the DNAs and the ADP ribosyltransferase, and that allowed those two to come together. And so that, I mean, that's, you know, amazing, right? And I don't think that there's any examples out there. There wouldn't be a logical reason to have two DNases. You know what I mean? They would do the same thing. That's a sort of big target, but it'd be really neat, for example, if there were two different ADP ribosyltransferases that had different targets on the cell or something. I mean, and you know, you dig deep enough in some weird organ, who knows, maybe that's out there, but we've never stumbled across anything like that. What we see are a tremendous diversity of delivery mechanisms, basically. And we see that we have these two enzymatic activities being delivered in all those different ways, but that's not to say that the different A subunits can't have different targets. So we find instances of the two different, so sequence different typhoid toxin islands that would have different PLTA sequences, right? Different ADP ribosyltransferases that might have different host cell targets, for example. Now, I don't know if they're being covalently tethered together. I guess it's not impossible, but we don't have any, I don't have any sort of evidence that suggests that's necessarily going on.
Yeah. So is there more interaction specificity between those two A subunits than just that disulfide bond? Like, or is it literally just a disulfide bond that's holding those two together?
It's by and large just the disulfide bond. There's some shape complementarity that happens there that allows that to happen.
Wow.
But if you disrupt, like if you mutate either of those two cysteines, that interaction is just completely gone. So, so, you know, sort of basically fitting together, I think, is important too. But yeah, and I mean, a big question comes up, right? Is that disulfide bond always there, or can you get toxins that only have the one, right? Sometimes you may not want the other one. And because they're regulated from different promoters, they're sort of transcribed from opposite strands, there's reason to think that maybe sometimes you're only making one of the toxins, or the AB5 instead of the A2B5.
Very cool. Thanks.
Thanks, Jason.
Okay, there's a couple in the chat, Casey. Can you read the chat, or do you want me to?
I don't have the chat open now. Let me.
I can read them to you if you'd prefer.
Let me open up the chat. There's some parasites. Good question. So Heather asks whether or not there are any sort of long-term human adapted lineages that sort of don't want to be, if I'm reading this question right, and Heather, please correct me if I get it wrong, sort of that aren't as destructive as typhi are, right? That sort of adopt more of a commensal role, that you know, sort of more of a friendly interaction rather than a pathogenic interaction. So we don't know of any in humans. There aren't any in humans. I think we probably would know about that. But I definitely think that that's going on in reptiles, for example. So I think that many of the lineages in reptiles and many of the interactions in reptiles are probably long-term stable interactions where it's more like a commensal. But with respect to humans, I don't think that that's evolved yet. Does that? How long has the relationship been going on? Oh, sorry, for typhi. So typhi's thought to be about 50,000 years old, I think is one estimate that I saw. So it probably evolved from, you know, some Salmonella out there, precursor, and it's been in the human population for about 50,000 years that's been going on. And then there's another lineage called Paratyphi A that's, I think, a little bit newer. I think that that seems to have evolved more recently, but still tens of thousands of years ago, essentially. Okay, so Martin has asked whether there's any evidence that the ADP ribosyl transferase in Salmonella targets G proteins. Great question, Martin. So you obviously know something about AB5 toxins, right? So I showed cholera toxin and I showed pertussis toxin, and both of those are ADP ribosyl transferases that target AG protein in the host cell. So that would be a great place to look. It's possible that that's going on. The primary person who's really, the primary lab that's really looked at that is sort of where I did my postdoc, is Jorge Galan. And he, so I've never kind of dove in on that angle, and no other lab that I know of other than the Galan lab has really sort of attacked that. And he's attacked it hard. And he's got this really, I actually love it about him, philosophy that that's not the way that he would go about that he wants sort of an unbiased experiment that shows that, rather than to say, can I show that this thing can target an ADP ribosyl transferase? He wants to say, I want to know what this thing targets in a physiological context. Can I figure that out with an unbiased experiment? And so because of that approach, it's been a real struggle for him to nail it down, right? But I think it's, you know, it's entirely possible that it does target a G protein, but I don't know is the answer to the question. It's a good guess, certainly. Does that answer your question, Martin? Sorry, I got it. Okay, great.
Okay. Do we have any more questions for Casey? If not, Casey, I want to thank you for giving really entertaining and very colorful presentation with balloon animals and Lego and Homer Simpson and all kinds of threads that you managed to pull together. Well done. I would also like to present you with your very own highly coveted Inspired Discovery for a Better World water bottle, which you will find in your mailbox sometime in the not too distant future. Next time you're on campus, maybe you can pick it up. What's that?
I haven't been over there, and I wonder how many things are in my mailbox.
I just had the same thought, to be honest. I was in today and I thought, oh, maybe I should check out my mailbox because I haven't looked at it for a year.
Yeah.
Yeah, I just had the same thought. So this will motivate you next time you're in there to actually poke your head in and see what's in your mailbox. Yeah, and with that concludes this year's series of Chair's Lectures. I want to thank all the speakers, and obviously thank Casey for doing such a great job today. And I'm sure this series will reinvigorate in the fall, and there will be a new lineup of speakers. I think it's been a fantastic thing for the department, and I know we've all been enriched by learning about what our colleagues do, and it's been, I think, good for building a sense of commonality and community in the department. I think it's a really good thing. It's a good thing for us. So thank you for everyone who's attended, and thank you for all the presenters. And with that, I will say good evening, and all of you take care. Hope to see you soon.
Thanks, Dave.
Thanks, Casey.